Okay, so far we've looked at monohybrid crosses and dihybrid crosses. Now we're going to add something a little different. It's called incomplete dominance. So far we've talked about the capital letters being the dominant alleles and the lowercase letters being the recessive alleles. In incomplete dominance, one allele is not dominant over the other. So what does that mean? That means we're going to get a mixture of the alleles. So one particular plant that has incomplete dominance are 4 o'clock flowers. So in 4 o'clock flowers, instead of just red or white, like we had with our purple flowers, for example, we had either purple or white. There was no in between. But with 4 o'clock flowers, because they have incomplete dominance, they can have this in-between category. What would that be? Well, mixed red and white, and a possible phenotype would be pink. So now instead of only 2 phenotypes, we have 3. So how are we going to show that? Well, notice that in our genotypes, so for red, big R, that looks familiar. But with white, we have still 2 big R's because it's not recessive anymore. So to show the difference, I put this little dash here, which we called a prime. We saw that when we had our 3' and 5' ends of our DNA strands. It's going to be the same way here, or I can show it as big W to show that it is white. Either one of those is fine. This one's a little more scientific, but as long as you get the right answer, it doesn't matter to me. Okay, so how would we get pink then? Well, we would either have big R with an R prime, or I can have big R, big W. Red and white make pink. So how would I do that for one of my crosses? So I'm going to cross a red plant with a white plant. So just like always, I need to list what those genotypes would be for their parents. So I have big R for red, just like we had over here. And I have R prime, got a little smudged, for white, just like I had over here. Again, if you had big R, big W, that's fine too. Okay, so I filled out our Punnett square for us, and we'll fill in the boxes just like we always do, bringing the top down and then whatever's on the side in. So I'd have big R, R prime in the first box, big R, R prime in the second box, and we see that they all end up being big R, R prime. So my genotype ratio, or my percentages, I'm looking at all 4 boxes are R, R prime. So I have 100% R, R prime. What about my phenotypes? If something has R, R prime, what is that going to be for the phenotype? Well, if I look over here in my chart, to remind myself, R, R prime is pink. So I have 100% pink flowers if I have a red plant being crossed with a white plant. Let's try another one. So this time I'm going to cross a red plant with a pink plant. So what would those genotypes be? Well, red is capital R, capital R. Pink is capital R, R prime. Or again, you could use RW, whatever makes more sense to you. I fill in my Punnett square, so I pick one of these. Doesn't really matter which one goes on the top. So I'll put R, R on the top, R, R prime on the side, fill in my boxes, R, 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 R prime, R, R prime. So what are my genotypes? Well, half of the boxes, so 2 out of the 4, or 50%, are capital R, capital R. The other 2 boxes at the bottom are R, R prime. So the other 50% is R, R prime. Okay, phenotype. What is that going to look like? Well, big R we discussed was red, so 50% red. The other two boxes are R prime. What is that? Pink. And we're done with that one. So at this point, I'm going to give you some examples to do on your own. You'll want to write these in your notebook. Give me a second to get the slides. Okay, so what I'd like you to do for these is to write all of this down and then do the problem on your own. When you come into class, we'll check them. So for this one, we'll notice that I'm not using the 4 o'clock flowers anymore, so kind of giving you a little bit of a challenge. Instead, I'm using hair. So I have, my phenotypes are, we could have curly hair, we could have straight hair, and then this last one, that would be the phenotype that's in the middle of those two. So what would be in between curly and straight? Wavy. So we can put wavy here. And then for my genotypes, I can use C's, C and C prime. I could use C's and S's, like we did with the R and the W. Either one of those is fine, whatever works best for you. So you'll take the same steps. You'll look at the problem, figure out the parents' genotypes, fill in the Punnett square, figure out your genotype and phenotype ratio according to what you get in your Punnett square. So now would be a good time to pause it and write down the problem in your notes and figure it out. Okay, problem 2, same idea, using curly, straight, and wavy. Go ahead and pause it and do the notes, or do the problem in your notes. And our third problem, still using curly, wavy, straight, and we have a new problem. Go ahead and write it out in your notes, and I'll see you tomorrow.